Pollution of the world is an ever-growing problem. Vast amounts of oil used to run vehicles, factories, and even power homes contaminate water in the air. The world has an estimated 1.3 trillion barrels of oil left in major oil fields. Scientists estimate those supplies to only be sufficient for only 40 more years. Hope lies in the most unlikely candidate, algae. Our solution was to stress the algae by using a variety of different light sources. We would then use a flow cytometer to test the lipid content. We would then see which light source made the algae produce more lipids. Whichever sample had the highest lipid content would be the best light used to produce algae biofuel. Chlorella is a single-celled green algae that is spherical in shape. Its diameter is 2 to 10 micrometers. It has no flagella, and it contains the pigments chlorophyll A and B. It multiplies very rapidly, which is good for reproducing it in a lab setting. Its average lipid content is 27%. The conditions to grow algae are very simple. All it needs for reproduction is carbon dioxide, water, light, and a small amount of minerals. There are many advantages to growing Chlorella vulgaris. It can grow in wastewater, and NASA's Omega project claims it does not compete with other agricultural activities for resources like water, fertilizer, and arable land. The Omega project is a project that researches methods of treating wastewater and producing biofuel. The purpose of our project was to expose Chlorella to a variety of different light sources in order to study the effect it has on its lipid content. We used red light, blue light, grow light, and ultraviolet light. Our experiment showed that the red light produced the highest lipid content at around 90%. However, it also had the highest transparency, which means the least amount of algae grew in it. The blue light had the third highest lipid content at 77.7%. However, unlike the red light at 73.3% transparency, the blue light only had 9.3% transparency. The grow light yielded the second highest percentage of lipid content at 79.7%, and it also had the lowest transparency at 5.3%, which means under the grow light the algae grew the most. The ultraviolet light had the lowest percentage of all our samples for its lipid content. It only had 27.4%, and it also had the second highest transparency percentage at 29.3%. Flow cytometry literally translates into the measurement of cells. It measures several aspects of the cell, including size, shape, fluorescence, and the makeup of the cell. There are several parts to a flow cytometer. The optics aspect of it is when the cells are analyzed by lasers. The electronic side is when the light is transferred into electrons, and these electrons are detected by receivers. There are several programs used to analyze the data received by the flow cytometer, and we used the computer software called FlowJo. We also did a ploidy analysis, and this was used to look at our fluorescence data. Here's some helpful flow cytometry vocabulary. Fluorescence is the light that is emitted from the fluorochrome or the dye after it absorbs the light. Scatter is the light that is deflected, reflected, or bounced off of a cell. Forward scatter measures the size of the cell. Side scatter measures the complexity of the organelles. The analysis point is where the laser illuminates the cell. The process of flow cytometry is fairly simple. There's a sheath fluid that surrounds the cells and aligns them for the lasers to be able to go through them. The sample core is the stream inside of the sheath fluid, which carries the cells in a single line for the laser, and the laser illuminates the cells and reads the data. There are many uses for a flow cytometer. It can be used for research like our project, medical diagnostic, marine biology, protein engineering, pathology, and it has several different lasers to detect several different types of data. So why bother with algae biofuel? The annual productivity and oil content of algae is far greater than any seed crop. Per hectare, which is about 2.5 acres, soybeans produce 450 liters, canola seeds produce around 1,200 liters, palm seeds around 6,000 liters, and algae produces around 90,000 liters. There are many advantages for using algae as a biofuel. It has many byproduct uses, such as being used in aquaculture feeds. The American Chemical Society stated that chemists' recent developments using chlorella as a biofuel has shown it to be the first economical and eco-friendly process that does not produce wastewater. The biggest advantage for using algae as a biofuel is that it greatly reduces the pollution in the air. During biofuel production, algae consumes carbon dioxide through photosynthesis. Algae pulls carbon dioxide from the air and replaces it with oxygen. For this reason, algae biofuel manufacturers are building biofuel plants close to energy manufacturing plants that produce a lot of carbon dioxide. It recycles the carbon dioxide, which reduces pollution. While there are many advantages to using algae as a biofuel, there are also some risks that come along with it as well, such as price. In 1996, it was still not economically viable to produce biodiesel from microalgae because, even when using best-case scenarios, a photosynthetic productivity price would still be twice as high as the price of a similar quantity of petroleum diesel. In stark contrast to oil prices of 1996, the price of petroleum today has more than doubled and therefore should be provided as an incentive for the creation of biofuel.